好，直播中可以播得啦。 So the size of the cell envelope, okay? So this is the size of the cell envelope, okay? So the size of the cell envelope, okay, and the cytoplasms, the regions, and the appendage. So appendage means, okay, the uh, the uh, the attachment, okay, on the surface of bacteria, okay. Sorry for the sound, okay. So see appendages, okay. So some bacteria do not have flagellum, or they have more than one flagellum, and some of the bacteria have another pilus, okay. So appendages mean the extra attachment on the surface of the bacteria, all right, okay. Right, so let's take a look for the cell envelope first, okay. Now, this is the size of the cell envelope, okay. So the size envelope, okay, the outermost shell is a capsule, okay, capsule, okay, which or the slime layers cover outside of the cell wall. Now, remember, the bacteria have cell wall, okay, but the structure of the bacterial cell was completely different from the plant cell wall, okay. And the inner layers of and the capsule just cover the cell wall, and the cell wall and cover the plasma membrane. So there are three layers, okay, of the cell envelope. Okay, so the cell envelope is composed of three layers. The outermost layers is the capsule, the cell wall, and plasma membrane. Now let's take a look for the capsule first. Okay, it's the outermost layers. It's a slam. Slam means sticky. It's a sticky layers. Okay, cover the cell wall. The slam layers is composed of the thick polysaccharide. So what is polysaccharide? Polysaccharide means the sugar. Is a sugar. For example, the starch, cellulose. They are the thick polysaccharides. So the primary function of SLAM surface is to help the bacteria to stick on the smooth layers. Okay, and sometime in starvation, when the bacteria do not have any food available, so they serve as a reserve, okay, for the food of the and energy reserve for the bacteria. And the cell wall, okay, the cell wall is to protect, okay, from the bacteria from mainly of the osmotic pressure, because the bacteria do not have the skeleton, they don't have the exoskeleton. So the cell wall, okay, provide a mechanical support. So what is a mechanical means like a wall, okay, to protect the bacteria from the stress. For example, the air, okay, some of the bacteria, they located underneath of sea or in the mountains, very high altitude. So they need to be, they're very stressed by the air pressure outside. So the cell wall is to protect the bacteria, okay, from the mechanical support, and of course the hypotonic means the water. So the difference of the level of water may stress them. So the cell walls provide a mechanical support. And finally, plasma membrane. So the design and structure of bacterial plasma membrane is similar, okay, to the human, okay, or other eukaryotic cell membrane, but there's some difference of their design, because bacteria do not have mitochondria. So the ATP synthesis occurs in the plasma membrane. Okay, so the primary function of the plasma membrane is to, okay, selectively or semi-permeable. Semi-permeable means they don't really wide open to the passage of in and out of the materials. They selected, they have the channels like a protein. So the plasma membrane is composed of the proteins and the fluid. Okay, right. So let's take a look for the capsule. So this is the capsule. Okay, so this is the white color. Okay, the white color of capsule. Okay, to stick together. Okay, they protect the bacteria from the. Now another functions of the capsule is because of slam. Okay, so they got a clove. Okay, on the surface of bacteria, so they can be. They try to avoid for the surveillance of the phagocytes. So phagocyte means a white blood cell to eat them. So they have to protect them. So they will stick together, okay, because the bacteria do not have any attachment. They don't have legs. They're not Spider-Man. So this lamb help them to stick on the smooth layer. So they will stick together one by one to form colonies. Remember, the bacteria is same as us, okay, human. We can't live together. We can't live alone, okay, in a lonely planet. We need to live together, okay, as well as bacteria. Bacteria can't survive as a single cell alone. They need to build up a colony. They need to build up a community, okay, to survive. They help with each other and to fight with their other types of bacteria. So they will grow, okay, because the speed of growth is rapid. For example, human cell division takes about 24 hours to complete, but for the E. coli, for example, only 90 minutes. So they can, okay, form together in large amount, okay, in very short period of time. So look at this. This is a turret. This is the plate on the tooth. Okay, this is a good examples. Okay, on the slim surface, so they stick together one by one. So look at the beautiful structure here. Okay, the plate. Okay, all right. So that's it for the capsule. Now we look at the cell. Now bacterial cell wall. Now, one of the way to distinguish between the types of different bacteria by looking at their size, the shape, okay, their living habitat, and their cell wall. So cell wall is one of the indicators to look at the types of bacteria. So generally, the bacteria is divided into two groups. The first group is the grand staining. So we stain it. So what is stain? We did the dye and color to look at the color of the surface of bacteria. So there are two colors, the purple or the pink color. So there are two groups of the bacteria, except for several of bacteria. So I will describe this later. Okay. So the gram state, the gram positive, okay, and gram negative. So gram, okay, gram is the method of the staining, okay. So why then? So because when the dye is added on the surface of bacteria, they form interactions, okay. So I'll show the video so how the gram staining is done, okay. So the bacterial cell wall is distinguished from others, okay. Fungus, plants, they have cell wall but different structure. The bacterial cell wall is primarily, okay, composed of the peptidoglycan, okay. So, but they have different things. When you look at the graph here, okay, the cell wall, there's a multiple layers of the peptidoglycans. Okay, it's much thicker than the gram negative. Okay, so the gram positive have a very thick, okay, polypeptidoglycans. Okay, however, the cell, so there are two layers. Okay, the peptidoglycans is in between the cell wall here. Okay, this is cell wall, and just on top of plasma protein. So plasma membrane. So there was sandwich-like structure. Okay, it's a pink color. Okay, they have outer membrane when it's given a dye. Okay, it's stated in the pink or red. And for the gram positive and stain it, so is a purple color. So that's it. Okay, the majority, okay, of the material to build up or constitute to the cell wall is the peptidoglycan. Right. So what is the peptidoglycan? So this is the structure. Okay, showing you is peptidal. Peptidal means a peptide. Peptide means a chain of the amino acid. Look at here how. So these are the amino acid, okay? So amino acid, they form a chain, okay? There's a bond, okay, to between the amino acid is said to be peptide bond, okay? So the peptide attached with the glycan. So glycan, okay, is the sugar. So the peptidoglycan means the chain of amino acid attached to the glycan. Glycan means the sugar. It's a mixture, is the combinations of the protein and sugar. So it's made of two types of sugar. Okay, so don't need to panic about the names, okay, of this sugar then, okay. So this is the sugar, the peptidoglycans and the two, okay, sugars, and then they form a linkage, okay, they form a very strong linkage, okay, so giving them rigid, okay, and so that can provide a mechanical, okay, strength and protection of the bacteria, okay. Right, let's take a look for functions of the bacterial cell wall. So bacterial cell is not only, okay, to provide a mechanical, okay, protections or give a shape. So they need to maintain shape. So that, so the shape made it us to distinguish them. The three basic shape, okay, have been seen in the world so far about the spherical shape, okay, cone or the rod shape, bacilli, or spiritual shape, vibro, okay. So the first function of bacteria can maintain its shape, okay, to maintain the integrity of the cell, okay, of the bacterial cell. And second, it protect from the water. So the water is a major, okay, that will kill the bacteria. So the osmotic hydrolysis to prevent the leakage of the fluid, okay, because of the difference between the cell and outside of the watery pressure or the osmotic pressure. And third, of course, the toxic material. So the bacteria, they live in and they adapt environment, so that sometimes some uncertain fluid can kill them, so they're toxic agents. So the bacteria cell will help them to protect from toxic material, and they constitute the pathogenicity. So we'll describe how the bacteria kill the host, or how to make the host sick because of the pathogenicity, because of, especially for the gram-negative, or the LPS, which is the endotoxin. So bacteria can secrete some enzymes or the toxin and make us sick. So the gram-negative, okay, of the structure can constitute to the pathogenicity, okay. So there are four functions of the bacterial cell wall, okay. All right, get it for the all right, so let's take a look for the gram-positive bacteria. Okay, so let's take a look for the gram-positive bacterial cell wall. Now, as I said earlier, okay, the bacterial cell wall is relatively thick. They are very thick, okay. So because they constitute, they're composed of multiple layers, a lot of layers, okay. So the positive bacterial cell wall is a very thick peptidoglycan, okay, and they stick it, so the plasma membrane here underneath of the peptidoglycan underneath, okay, so they have a stick, it's a stick-like structure to hold them together, okay. So tiotic acid, okay, ticotic acid, okay. So the particular means they are straight, vertically, okay, penetrate through the peptidoglycan, okay, the liponucleic acid. So they help to hold and bind to the cytoplasmic membrane, okay. So they help to hold it, okay. So to attach, okay. So that can maintain, okay, the cell envelopes, okay, and of course to protect from the environmental substances, okay, and they help to bind, okay, with the host cell. They help them to stick the, okay, cell on the stick, okay. Okay, any question here? So so the break will be given in a certain time. So don't worry. Uh, so don't worry about this. Okay. So there is a question time. Okay, later. Okay. So you can add. So if you have any question, you can add a chat. Okay, in the right hand side of the YouTube. Okay. So there is a sound here. Okay. So the sound shouldn't have any problems because I didn't turn off sound in the beginning. So the sound should be okay now. Okay. Right. So this is the like so generally speaking, okay, the gram-positive chart, gram-positive bacteria is and the bacteria wall, okay, looks like a cheesecake. So you can take a cheesecake after the lecture today. So this cheesecake, a cheese looks like a multiple layers of the peptide ligands, okay. So at the chaotic acid, okay, serve to maintain the cell shape, okay, and the lipochaotic acid, okay, is important cell wall, okay. Right, so that's it for the gram-positive bacteria, okay. Now we're looking at the gram-negative bacterial cell wall, okay. So gram-negative bacterial cell wall, okay, in which the, the thickness of the peptide ligand is much less, okay, than the, okay, that of the gram-positive bacteria. Right, so this is so the bacterial cell wall here, okay, is in the middle between the outer membrane and the inner membrane. So they form a three-layer structure, like structure. And the second difference is there's no chaotic acid found. Okay, there's no chaotic acid found on the gram-negative bacterial cell wall. And the outer membrane is composed of the lipids. Okay, so it's a lipids by layers. So there are two layers of lipids, and there's a unique structure found in the gram-negative bacteria. That means the lipopolysaccharides, okay, LPS, is only found, okay, in the gram-negative bacterial cell wall, okay, and lipoproteins. So lipoproteins, okay, and the LPS, okay. So what is LPS? So LP, lipo means is a lipids, and polysaccharides is a combination of the lipids or fats. So lipid means a fat, okay, and sugar. It's a lipid sugar. It's a lipo sugar, okay. So it's composed of three parts. The lipid A here, so the lipids, okay, the core. So the core means the core polysaccharides, the multiple sugar, and the O antigen. So antigens, okay, is found on the negative, okay, gram bacteria. So these O antigens, okay, is something like the trigger. So when the negative gram bacteria, when they invite our human body, okay, can recognize the O antigens, okay. So this is the general features, okay, of the lipoproteins. So what they do then, okay, so of course the first function is to maintain the shape, okay, is to protect, okay, to stabilize or integrity of the cell structure, okay, and second, make the constitute to attach. So they need to attach on the surface of the host, okay. And the third thing is to create of course, it helps to create a barrier to selectively for the in and out of the materials, okay, to avoid the toxinization, to avoid the death of the cell, okay. And fourth of the O antigen is to protect from the host defense, and lipid A here is the site for secretions of the endotoxin. So on the other hand, about 95% of the gram-negative bacteria, they're pathogenic. Now, what is the meaning of pathogenic? It means they're toxic because of they can secrete the endotoxin, okay, that harm us, okay. All right, so any question here? So you can type any question here on the right-hand side of the instant chat, okay. So I can answer any question. So the sound is okay. Thank you. Okay, the sound is okay now. Right, so this is the gram negatives, okay. So it looks like sandwich. So in the middle, yummy, okay, it's a cheese, okay. So the polypical ligand is a cheese, okay. So this is covered by the the outer and inner membrane, okay, the cytoplasm, the plasma membrane and outer membrane, okay. Right. Right, thank you for the question here. Yes, the live can be saved. Okay, the live will be saved after the lecture today, so you can replay it. Okay, so for the whole week, and then I'll provide the link and I'll put it on Soul. Okay, because after a week, this lecture will only be seen. Not open. It's just for the student of our MHPM student. Okay, so again, the lipopolysaccharides is a major features. Okay, LPS is only found in the gram negative, and the lipid A will create a endotoxin, so make it toxicity of the gram negative bacteria. Now, on the other hand, most of the gram positive bacteria are not pathogenic. About 9% of the gram-positive bacteria are non-pathogenic. However, 95% of the gram-negative bacteria are pathogenic. So again, this is a summarized table. It's a table to summarize the differences between of the cell wall between the gram-positive bacteria and gram-negative bacteria. So again, the first difference is the thickness of the wall is much thicker, than the gram-negative bacteria, And secondly, the components of the cell wall, the ketotic acid, However, for gram-negative, only found is the LPS, It's a very complex structure. So the LPS is the major component is found in the gram-negative bacteria. On the other hand, the ketotic acid is found in the gram-positive bacteria. So any question here? So if you have any question, you can ask me through the YouTube online here, YouTube on the chat. Or you can ask me through the Soul, the chat of Soul. So you may add me the chat of Soul today. So if there's no question, so this is the cell wall. So this is strategy. So because of cell wall, sometimes, as I said, not most of the gram-negative bacteria and 10% of gram-positive bacteria are harmful to humans. So we need to find a way, because bacteria can kill us. So we need to find a way to against the bacteria, to kill the bacteria. So how we do that? So as I said in the first lecture, the Alexander Fleming in 1925, so he accidentally discovered the penicillium. So how penicillium works? So when you look at the graph here, so this is a graph. All right, so this is a graph. So the graph here. So look at here. This is the penicillium. So penicillium is a group of the beta-lactamates. Of the antibiotics. So antibiotics means they against the bacteria. So how the penicillium works by inhibits the cell wall synthesis. So the cell wall, they need to regularly maintain a shape. So the penicillium is to inhibit the synthesis of the bacterial cell wall. So that means that the bacterial cell wall can't be maintained. That's the graph here. So this is a graph. So penicillium, it works quite straightforwardly. So they will inhibit the breakdown. They break down the cross-link. So this is a cross-link. The peptide ligand. So they inhibit the synthesis. So the cell wall can't be maintained, and that means and eventually the cell wall lost the integrity of the bacteria, and eventually the lysis. So eventually the bacteria is killed by the lysis. But however, the bacteria can secrete antibiotics, can secrete something to break us, to protect them. So the second generations of the bacteria, they learn the hard lesson, because the parasites or the son or the offspring of bacteria, they learn it. So how they learn it? So they secrete the enzyme, the bacterolactamase. So this is an enzyme. They break the penicillium ring. So when we take the penicillium, the bacteria will secrete an enzyme to disable, to break, the ring here. This is a ring, the beta-lactamine ring. Okay, so they cause the dysfunction the whole molecules. Okay, so it can't work then. The bacterium can't work anymore. So this phenomenon is said to be antibiotics resistant. So the bacteria can resist the actions of antibiotics, okay, by secreting an enzyme that disable or to destruct the structure of penicillium. Right, so at this time, let me show you a video, okay, on about the antibiotics resistance, okay, so in animations, okay, so you can, and I will upload this video, okay, on YouTube, so you can watch it on YouTube in your lecture time. So let me play the video now.
Bacteria are primarily classified into two main types, gram-negative and gram-positive. This designation is based on a laboratory test that distinguishes bacteria based on the composition of the outermost part of the bacterium, known as the cell wall. Overall, infections caused by gram-negative bacteria now account for more than 30% of common hospital-acquired or nosocomial infections. In addition, they are the leading causes of nosocomial pneumonia and urinary tract infections, or UTIs. Gram-negative bacteria such as Pseudomonas aeruginosa, Klebsiella pneumoniae, and Escherichia coli are becoming more resistant to the antibiotics we have available, leaving doctors with limited options to manage patients with infections caused by these organisms. Gram-negative bacteria have a cell wall that lies between the outer membrane and the inner cytoplasmic membrane. The cell wall contains penicillin-binding proteins, or PBPs. PBPs help build new cell wall during growth and division, maintaining its integrity. Beta-lactam antibiotics bind to these PBPs and weaken the cell wall, resulting in the cell's death. Gram-negative bacteria, common in hospital-acquired infections, often contain several mechanisms that can render antibiotics ineffective, including changing the outer membrane and preventing antibiotics from entering, and pushing antibiotics out that do get through by using efflux pumps. Certain gram-negative bacteria can also produce beta-lactamase. These enzymes inactivate beta-lactam antibiotics and keep them from binding to PBPs. Some gram-negative bacteria can produce powerful beta-lactamases called extended-spectrum beta-lactamases, or ESBLs. ESBLs can render penicillins, cephalosporins, and monobactams inactive. In order to effectively treat infections caused by drug-resistant gram-negative bacteria, new antibiotics that overcome multiple mechanisms of resistance are needed.
Okay. So again, sorry. Okay. So the sound here. Okay. Right. The sound. Okay. The sound is okay now. Sorry. So I will repeat again. I will repeat what I said. Right. I should be fine now. Oh, sorry. Okay. Okay. Let me just. Okay. Right. So sound is okay now. So I repeat what I said. Okay. So the cell wall synthesis. So the antibiotics. Okay. One of the way is to inhibit the cell wall synthesis. But as I said, okay, some of the bacteria they find a way to reject them. So the scientists they continuous to find a way to inhibit the activities of the cell by block the DNA or block the translation. So in other groups of antibiotics, they inhibit the cell divisions or to inhibit the bacteria to produce the proteins that are essential for the metabolisms. So the bacteria eventually died. But however, the bacteria is smart. So they continuous to resistant the drugs then. So the scientists you need to have a mission to continuous to create some antibiotics. So that's why the doctors won't recommend the patient to take too much of antibiotics because sometimes some patients they have multiple of the attack of bacteria, but no, the antibiotics have failed to treat them. Okay, so that's it for the about the additional information about the inhibitors of the cell wall. Right, so let's take a break. Okay, so let's take a break. Okay, for 10 minutes break. So this is a Q&A session. So any of you have any questions, so you can ask me. So we will come back at 10:20 about 10 about 11 11 20 So we come back at 11:20 then. So if you have any question, you can ask me during the break. So I play the music. That's my favorite music. So enjoy music. We come back 11 20. Okay, so welcome back to the second part of the lecture today on the lecture two, the structure and the functions of the bacteria. All right, so the chat is open on the right hand side. It's open for the questions. Okay, if you have any question, you can type okay on the instant chat. Okay, right. So let's continue the lecture. Okay. Right, so the second, okay, the components of bacteria is the appendage. Appendage means the attachments, okay, on the cell wall, okay. So attachment means, okay, some of the bacteria do not have the of the flagella, okay. Some of them they have multiple, one or two. So this is another way to classify the types of bacteria. All right. So the first one is the flagella. Flagella means the like a tail. Is a long curved tail. Okay, some of the bacteria are in some of the flagellum is absent. Okay, on the surface of the bacteria. Okay, so the flagellum is to help to localize, is to locomotion, to swim like a swim. Okay, of bacteria. Okay. Right, and this is one. Okay, so I will play a video. I'll show you a video about how the flagella works. How the flagellum works. Okay, so I'll give you a video. And the second one is a pili. Now these are pili. Okay, now look at here. These are the pili. Okay, So this pili, okay, are short protein. So they're protein stick-like, okay? It's like a hair, okay? It's a hair-like structure. So it helps to fix it, to help to fix the pili on the wall. So the bacteria has, they have the slime layers, okay, the flagellum, so the pili, that helps the bacteria to stick on the smooth, okay, layers, to stick them well. Okay, and the final one is a primate, the fin blade. The fin blade is also known as is attachment pili, okay, and they helps to get avoid for the surveillance of the leukocyte. Remember, leukocyte is the first lecture on white blood cell, okay. So the white blood cell they can eat, so they can eat the bacteria. So the bacteria has the pili to get avoid, okay, so they can cover them to avoid the surveillance, okay, of the bacteria, okay. Right, so let's take a video on the, so this is the structure of the flagellum, okay, is made of a motor-like structure. So actually this is an ATP synthase. It's a hydrogen pump. Remember the mitochondria, okay? So the mitochondria, so they are the complex, okay, on the mitochondria for the generation of the ATP. So in the complex 5, there's an ATP synthase when the proton, so they were driven from the outside to inner, so they will rotate the motor, okay? So the flagellum, they can work, so they will turbulence, like a turbulence, okay, of the way, okay? So that the bacteria can mobilize, okay? Okay? So let me play the video of the mechanisms, okay, of the flagella.
Many species of bacteria propel themselves through their environment by spinning helical motorized flagella. Rhodobacter cells have one flagellum each, whereas E. coli cells have multiple flagella that rotate in bundles. Each flagellum consists of a helical filament that is 20 nanometers wide and up to 15 microns long, and spins on the order of 100 times per second. These animations show a series of schematized and speculative models about how bacterial flagella might function and assemble. Just outside of the cell wall, the filament is connected to a flexible rotating hook. The filament, the hook, and a structure called the basal body, located below the cell's surface, make up the 3 parts of the flagellum. The basal body consists of a rod and a series of rings embedded in the inner membrane, the peptidoglycan layer, and the outer membrane. Some of the rings make up the flagellar motor, which can be divided into 2 major parts: the stator, which is attached to the peptidoglycan layer, and as its name implies, remains stationary, and the rotor, which rotates. The motor derives its power from a proton gradient across the membrane. In this example, a high concentration of protons exists outside, and a low concentration exists inside the cell. The protons flow through the interface between 2 types of proteins, called mote A and mote B, that make up the stator. Mutational studies suggest that a conserved aspartic acid in mote B functions in proton conductance. Each stator contains 2 mote B proteins, and therefore also contains 2 of these important aspartic acids. Although the molecular mechanism of rotation is not known, one possible model describes protons moving through the channels in the stators and binding to the aspartic acid in the mote B proteins. This binding causes a conformational change in mote A proteins, resulting in the first power stroke that moves the rotor incrementally. At the end of the first power stroke, the 2 protons are released into the cytoplasm. The proton loss causes a second conformational change that drives the second power stroke, once again engaging the rotor. Although the mechanism for motor function is not yet certain, many details of flagellar assembly have been determined. Flagella begin their assembly with structures in the inner membrane. 26 subunits of an integral membrane protein called Fli-F come together in the plasma membrane to form the MS ring. The Fli-G proteins assemble under the MS ring. Fli-G, along with Fli-M and Fli-N proteins, make up the rotor. Flagellar proteins destined to be part of the extracellular portion of the flagellum are exported from the cell by a flagellum-specific export pathway and assembled at the center. Moat A and Moat B form the stationary part of the flagellar motor, the stator. Both are integral membrane proteins, but Moat B is also anchored to the rigid peptidoglycan layer, keeping the stator proteins fixed in place. The subunits of the rod portion of the rotor move up through the hollow cylinder in the assembly, and assisted by cap proteins, build up the rod in a proximal to distal fashion. Another set of rings, called L and P rings, are found in gram-negative bacteria such as E. coli. They penetrate the outer membrane, forming a bearing for the rod. As the rod cap is exposed outside the L ring, it dissociates and is replaced by a hook cap that guides the assembly of the hook proteins. After the hook is assembled, the hook cap dissociates, and a series of junction proteins assemble between the hook and future filaments. Finally, yet another cap is built, and filament proteins assemble. Like the rod and hook proteins, they travel through the hollow channel inside the filament to reach the distal end. The cap rotates, which causes the subunits to build in a helical fashion. A complete filament can consist of 20,000 to 30,000 subunits.
Oh, I'm sorry. Okay. So this is the way. So this is the way. Of the flagella work. So this is a structure. It's similar. To the to the humans one. So the hydrogen pump to pump the protons out and come back again to generate the ATP. And the hydrogen pump, so hydrogens, they will going down here through the motor. So the motor, will to turn the motors and eventually the flagella can rotate. So they can rotate because the motor, so the hydrogen pump inside and rotate. This is rotation. So this is a way how the bacteria generate ATP because the bacteria do not have mitochondria. So this is how they generate ATP. Right, so that's it. So we have, I have taught you about the cell wall, the cell envelope. Okay, the cell envelope contains three layers: the slime surface, the capsule. The second one is the cell wall, which is the most important components of bacteria, and the inner layer is a plasma membrane that's selectively for permeable of material. And appendage. So there are three appendage: the framing, the flagella, and the pili. Now we'll go on the third one, the cytoplasmic regions, the inner part, okay, of the cell, the cytoplasm. Of course, cytoplasms. So all the metabolism replications occur in cytoplasm because of two. Okay, the ribosome. So ribosomes is the machines, okay, for the translation protein. So it's a site of protein synthesis. Okay, and plasmid. Now, plasmid is the additions genes of the bacteria. So they a small circle. So there are two sets of DNA. The first one is the cytoplasmic the chromosome. So this is the this is a bacterial chromosome. So they appear, they can be seen, okay, on the cytoplasm. So they're loose in form. They are not structurally stable than humans one. So humans, so they have a nucleus. So DNA is inside of the nucleus. So make the DNA very stable. But on the other hand, the bacterial genes are less stable, so they can manipulate. So we can do the DNA recombinant technology. So that's why it's easier for scientists to do the DNA recombinant, okay, on the bacteria because they do not have nucleus, okay. And this one additional small plasmid. So this is where we can engineer, okay, the DNA. So what's the functions of plasmid DNA is for defense. So as I said earlier, so the antibiotics. So the bacteria can secrete the antibiotics, the enzyme to degrade our antibiotics by the plasmid DNA. Okay, so I'll tell you how they work then. Okay, so this is the general pictures of the metabolism. Okay, in the cytoplasm of the bacterial cell. Okay, almost all metabolism occur in cytoplasms. Okay, now plasmid. Now plasmid is a double-strand DNA. It's a small, very small. Okay, so they can existently have they work independent, okay, of the cytoplasmic chromosomes. Okay, and the first function is to provide the antibiotic resistance. So they can secrete the enzymes, the molecules that resist the action of antibiotics. So they can build an antibiotic resistance to against, okay, the foreign toxin, and capable to produce the enzyme and toxin to kill other bacteria. Okay. So this is the plasmid engineering. So on the left-hand side here is the map. Okay, so we can insert, okay, the genes here. Okay, we can insert the antibiotics here. So the bacteria can grow in the presence of the antibiotics. Okay, and this is the engineering of the plasmid proteins. So in general speaking, so there are two sets of the DNA. One set is the bacterial chromosome, another set is about the bacterial and the DNA plasmid. Okay, so let's watch a video on the introduction of the plasmid.
Bacteria have been helpful to genetic engineers, not only in providing them with restriction enzymes, but also with what are called plasmids. In addition to their single chromosome, many bacteria also contain tiny rings of DNA called plasmids. Plasmids are usually about 1,000 to 100,000 nucleotides long and act as independent, self-replicating molecular operators within bacteria. Plasmids are one of the ways that DNA recombination goes on within nature. While a bacteria's chromosome contains all the necessary genes to code for the bacteria's existence, plasmids provide an effective way by which traits not contained in the chromosome can be passed from bacteria to bacteria. For example, Some plasmids contain genes that code for enzymes that digest certain antibiotics, such as penicillin or ampicillin. This is obviously an advantage to the bacteria. When a bacteria containing these plasmids dies, it breaks open and liberates these plasmids to the outside environment, and they are often taken up by other bacteria that then acquire the traits coded for by the plasmids. Genetic engineers can place liberated plasmids in a solution with restriction enzymes and cut them apart. The engineers then place the gene segment they wish to insert into the solution with the plasmids, and then add repair enzymes that join the pieces of DNA together at their sticky ends. These new plasmids, with their newly inserted genes, are then placed with bacteria into a different solution that enables the plasmids to readily penetrate through the cell wall and membrane of bacteria. The bacteria that are exposed to the plasmids are taken out and grown in a culture for a short time. If the plasmids into which the experimental gene, such as the gene for the production of insulin or growth hormone, is inserted, also have genes that provide resistance to ampicillin and amoxicillin, a unique technique can be used. Genetic engineers know that not all the bacteria exposed to the plasmids will absorb them. By exposing the cultured bacteria to antibiotics such as ampicillin and amoxicillin, they kill off the bacteria that didn't absorb the new plasmids, while those bacteria that did absorb the new plasmids containing the genes for antibiotic resistance and for, say, insulin or growth hormone continue to grow and thrive in the culture. Scientists continue to grow these bacteria until the insulin or growth hormone produced by them is sufficient to be extracted and purified for use by human patients. But how are genes inserted into the chromosomes and DNA of more complex organisms such as plants or animals? There are two major methods of placing DNA into the nuclei of eukaryotic cells: vectorless and vector methods. Vectorless methods of insertion of DNA don't require the use of a third intermediate organism to deliver the DNA from one species to another. Vectors, in the biological sense of the word, are organisms that carry the DNA of one species and insert it into another.
Right, so this is a video shows you about the DNA recombinant technology. So let's take a graph here. Okay, so this is a graph for biotechnology works. So this is the cell, okay, taken from DNA, for example, insulin. So insulin is a good example because insulin is the first human synthetic human-made insulins. Okay, so how they work then? So we take out the genes, okay, sequence, okay, so the genes read the protein insulin. So we cut the genes of the insulin here and take it out, and then we'll insert. Okay, so we use the restriction enzyme. So this is the plasmid. So restriction enzyme, we cut it wide open. And then, so this is an insulin, they plug, so insert, okay, the plasmid, okay, and the plasmid, we put it back to the bacteria, so what they can read, the insulin proteins, okay, and this is how insulin is made, okay, from the plasmid engineering, okay, right, so now, that's all about the general structure of the bacteria. Now we move on the classification of bacteria. Okay, so there's a multiple ways to classify the bacteria. Okay, so bacteria is generally classified by three. Okay, the three groups. The first group by the appearance, the morphology, how they look. They look beautiful, they're beautiful or ugly. The shape, it means shape. The shape and the presence and numbers of the flagella, because I said some bacteria, they have one or two or multiple flagella, but the flagella is absent in some kinds of the bacteria. And pili, now there's two types of pili, the positive and negative pili. The negative pili, if you see in the video earlier, so these negative pili, okay, they found in the negative strand bacteria. They serve the nickname of the negative pili is the sex pili. Okay, so I describe the pili later. Okay, and the second one is a gram-positive. So this is a big part. So most of the microbiologists, they use the gram staining methods to classify the bacteria into the positive and negative strands. So by looking at the characteristic of the cell wall. And third, by looking at their habitats, the respiration. So some bacteria, they need to live in the absence of the oxygen, but some of bacteria, because for example, for E. coli inside the body, they are insufficient oxygen, so they can uptake a very small amount of oxygen. So they're flexible for the different concentration of oxygen. But some of the bacteria they can't survive in the presence of oxygen. They need a oxygen-free environment. Okay, and some of the bacteria of the food they taken. Okay, some of bacteria. Okay, they need to feed, and how they reproduce. Okay, the reproduction mode of the bacteria. Okay. Right, so this is the map, okay? So this is a map showing you, okay, the classification of the bacteria. So this is step. So step number one, they look at shape first. So they will look at the shape. So this is how we name the bacteria, by the shape, okay? So the coli means the round shape, okay? The spherical shape or the bacilli. And secondly, we stain the bacteria. So they're gram-positive or gram-negative. And third, looking at their adequates, how they need the oxygen levels. Okay, I'll describe later. And how they form a spore. And of course, the modern classifications based on their genes. So we do a gene sequencing. So how the virus then? For example, the coronavirus. So how we know the types. The Barrymore classification. So one of the technique by looking at the genes. So for example, let me just show you about the virus examples or instead of bacteria. So virus, they have the different classes of their genes. So DNA virus, the RNA virus. So coronavirus is about the type 4, the positive strand, single strand RNA. So we can do the DNA sequencing to looking at their subtypes, for example, type A, type B, type C, and type D, or new strand, etc., as well as the bacteria. So we can look at their biochemistry. So enzymes, some types of enzyme only secrete from one certain groups of the bacteria. So we can do a fast test. Some of students, they did a microbiology group, the group project, which disappointed me last semester. So I asked them to find a quick test. So they add a drop of the chemical, so they have a color. So this color represents some enzymes produced from bacteria. Okay, so we can look at their biochemistry or genetics, the genes, the DNA. So this is much more accurate, it's much more fast than the previous classifications. And serology typing. So we do the typing serology, look at the species. So this is here is the species, okay, of the bacteria. So we can group them as a specific features. So why we need these classifications, okay? For example, when you get sick, okay, so when you're really sick, okay, so we'll get you to hospital and they will uptake the fluid, the secretion or blood test. They do a blood test to serology. They do a blood test. The important blood test is to find out whether the bacteria is present in the blood. Okay, if yes, so they will culturing the bacteria, okay, and then look at their structure and look at under microscopy and do the test or take out for gene sequencing, so they can accurately find out a specific stand, the species of bacteria, and the doctors or the physicians or residents, so they can prescribe the appropriate of the antibiotics. So this is really important. So classification is really important for diagnosis and for treatment, okay, of the patients with the bacterial invasions, okay. Now, this is examples of classification. So let me just give you examples here. So for example, when it's stained, so we stain it, also it is a purple color, purple or very dark blue color. So obviously, this is a gram-positive strand. And then we look at the scope, it's a round shape. So round shape is a coli, okay, and this shape, okay, so this one is a bacilli, okay, bacilli, or the, right, let me take out here. So this is a beautiful one. This is round shape. Okay, round shape is a coli. Coli means the round shape. Okay, look at here, the round shape of bacteria. Okay, so this is a round shape, and then we're looking at the respiration. So aerobic means they need a full amount of the oxygen. Okay, about the as air. Okay, or absence. Okay, and how shape. So some of the in the cluster form. So there are multiple of the cluster. Cluster means it's a great structure or chains or pairs. So we can name them several colas. Colas means the round shape. Several, the several means a cluster. It's a cluster form of the colas, okay, or chain from several coli. The several means a chain. It's a chain of the circle. They join together to form a chain. So we can name, okay, the genus and then S for the species because they have difference of the family members, okay. So that's it. So I will introduce you some of examples. So these are the pathogenic bacteria that I'll introduce you next lecture. So lecture 3, I'll introduce you some of these bacteria, okay. Now, so there are 3 basic shapes of the bacteria. So the first shape, okay, the first one is the colus. Okay, colus means a circular form. So this is circular form. Okay, colus. Okay, colus is a circular form. Okay, and the second one is a rod shape. The rod shape is a bacilli. So this is rod shape. Okay, or spiral shape. The spiral shape. Okay. So there are 3 shapes. Now let's take a look. Some of bacteria they form in the double form, the diplopoly. Diplo or diplopoly means the 2 strand. It's a double cone, the ice cream form. Okay, the double cone. Okay, so double. Okay, double. Okay, double. Okay, double. Okay, double of the spherical shape. And the second one is a chain form. The chain form, okay, several mean the chain form of the coli, or they form a tetrad. Tetrad means 4 in groups, okay, or the separate coli, the cluster form. The cluster form, okay, so there are different arrangements, okay, the shape and arrangement. And second one is the bacilli, okay. Bacilli, it means a chain of the bacilli or single one, the rods. Okay. And finally, spiral, fibrils. I'll introduce you to fibrils later. Okay. So these are the ones, okay, which is called just a series of diarrhea. Okay. So that's about the shapes, okay, and arrangement. Okay, clear? Now let's look at it here. Colors, okay. So these colors, okay, are divided in a one plane, only one plane. This is colors. Colors is one plane, okay. So this is the Diplocolus, okay. Neisseria. So Neisseria is one example. I'll introduce it to you, and all in 2 planes, okay. 2 planes, 1 plane, 2 planes, okay. Tetroid or 3 planes, okay. The octet and the random plants, the random shape of the colors. So there are 4 common, okay, of the shapes and arrangement of the coli, okay. Now let's take a look for the Neisseria. Now Neisseria, now this is a profile, okay. So give you examples. These are profiles of the bacteria. So this is gram-negative. So it looks like a pink color. So we stain it. It's a pink and red color. Now the flagella is absent. No, it's a non-motor. Motor means they can't move. They can't move. Okay, and they are diplocolli. Okay, diplocolli. Diplo means a 2 shape. 2. Okay, 2. Okay, there are 1, 2. Okay, and there are 2 pathogens. Okay, pathogens. Okay, the most serious, of course, this is the sex. Okay, disease. Okay, and that will and this another type is meningitis. The meningitis means they will cause you the inflammation of your brain. So there are 2 groups. Okay, when you look at here, it's a diplocolli. So this is a common of a sexual transmitted disease worldwide. Okay, And the second one is a Neisseria meningitis. It's second course of the meningitis in adults. So will cause you the inflammation of the human brain. Right. So let me just play a video, okay, of the Neisseria. Okay, just hang a minute. All right, sorry, no video, so I will upload the video on Soul later, okay? Right, so let's take a look here. Okay, now the second type is the bacilli. So bacilli means a rod shape. So this is a rod shape of bacilli. So single form, okay, the monobacilli example, say E. coli, okay? Second one is a dipobacilli, okay, pneumonia. So this bacteria will cause the inflammation of lungs, okay, pneumonia, and the cephalosporin is a chain form, okay, it's chain form, okay. So I'll introduce the E. coli second and the third lectures, okay. Right, so that's it for the shapes, okay, and the shapes, okay, arrangements of the bacteria. So let me just introduce you one more, okay, spiral. Now spiral means the vibrio, okay, the curve-like shape. Okay, so this is one, okay, commonly, okay, found in the Africa and the middle of the India, okay, so the Vibrio coli, okay, Cholera. So this Cholera could cause you the inflammations, or it cause you diarrhea intestine. So this is the one example, okay, the Vibrio coli, so this is a gram-negative again. So as I said, it's about 95%, okay, of the bacteria, of the gram-negative bacteria, they're pathogenic, okay. So the spherical shape, okay, and cause the intestine inflammations, epidemic, okay, or pandemic. Epidemic means it can be spread locally and spread worldwide, okay. That's cross, okay, the continent, okay. So this is one of the challenge because usually they cause a serious diarrhea and die of the dehydrations. Okay, usually die of the dehydration. So how they affect the body? Okay, so they mainly spread through the fluid. So it's a fluid. Okay, it's a fluid. Okay, contact. So we drink the dirty waters. Okay, because they're usually cholera found in the stool or in the feces of the patients, and because of the hygiene problem, okay, so the people they won't wash the hands, and that get the water and they contaminate food and they drink it or swallow it, okay. So these bacteria they live in the, so they will start their divisions in the intestine, okay. So this intestine they will cause, okay, the fluid, okay, the fluid flowing back, okay, from blood to the lumen. So this will cause the fluid, okay, and eventually severe diarrhea, okay. So that's it. So that's the mechanisms of the cholera, okay. So let's take a 10 minutes break. So we come back at 12:10. Okay, thank you. Right, so let's come back to the final section of the lecture. Okay, so right, so the before lectures, so this is spiral, okay, of the shapes of the bacteria, the third shape, third type of shape. Okay, so so I show you the video of the verbal of the story. Okay, it's about the story of this bacteria as I described it before the lecture. So let me play the video. Okay, now.
This is the story of how cholera changed my village. Tiny germs of cholera, too small to see, spread through the river. So small, yet so dangerous. Without realizing, women carried cholera home in the water. Flies carried cholera on their feet. Unwashed hands spread it too. We swallowed cholera germs in our water. on our food, and on our fingers. It happened so fast. By morning, my father was very sick. He had diarrhea that looked like gray water and poured out of him. I was so scared. I went for help. I never rode so fast. One look at my father and the nurse knew it was cholera. We had to work fast to save him. We made a special drink to help him. First, we made the water safe. We filtered it through cloth and boiled it for 1 minute. Then we mixed half a teaspoon of salt and 6 teaspoons of sugar in 1 liter of this safe water. It tasted like tears, not too salty. I worried my father would die before my eyes, but he soon felt a little stronger. The nurse explained to me that not everyone who swallows cholera germs gets sick like my father, but they can still spread the disease. Now I needed to take safe water to my village and teach them how to protect themselves from cholera. I saw a girl carrying water. I told her she could make the water safe by adding chlorine drops and waiting half an hour. There was a man about to eat with unclean hands. I told him to always wash his hands with soap and safe water after going to the toilet. Only with clean hands could he eat safely. I saw villagers spreading cholera into our river. I told them we needed to dig latrines far from the river, at least 30 meters away. This was important to keep our village clean. I found a mother preparing unsafe food. I told her, first, we must wash our hands with safe water. Then, we had to wash and peel the food, cook it and always eat it hot, and protect it from flies. I spread the word throughout my village and ran to find my father. I was so happy to see he was better. Our village became healthy. Now we filter and boil our water to make sure it is safe. We always use latrines and always wash our hands after. Food is safe from flies, washed and peeled and cooked. And we always wash our hands before cooking and eating. We made our village safe from cholera. Spread the word. Your village can be safe too.
So this is the video. I'll show you how importance of the personal hygiene in prevention of the bacteria. It works for the viral attack in Hong Kong now because the personal hygiene is a big contact. Okay, now we're going back to the another type of classification is the flagella. Okay, the numbers, absence and presence of flagella and the numbers. Okay, so there are 4 categories, okay, about the distribution, okay, about the distributions of the flagella. Okay, the first one is the entire surface, so it looks like a lot of the flagella, okay, enclose all the okay bacteria. Okay, so they are the peri-curious bacteria. They have a multiple flagella enclosing all the surface of bacteria. And second one is the lipo, okay? Lipo means there's a cluster of flagella on one end, so only one end. So this is the flagella, okay? The flagella. This is the flagella here. Is only one, okay? One figures of the flagella. So only this is the end, okay? The lipo, the end, okay? Is a lot of cluster of the flagella, okay? And the third one is a amphitriosis flagella, and amphiterius. Amphibian, amphi means both ends. These are both ends of the flagella, okay, of the ends, of both ends. So one and one, okay? And the final one is the mono. Mono means one, okay? Mono means one single, okay, of the polar, okay, of the flagella. Okay, so there are four types of the flagella. Okay, the flagella enclosed over the bacteria is a bacteria, the lithotritulus bacteria, amphiterius bacteria, and monoterius bacteria. Right, the pili. Now, there's two kinds of pili, okay, on the surface of bacteria. One is a common pili. So common pili means the commonly found in both of the gram-positive and gram-negative bacteria. Okay, so they are responsible for attachments of the bacteria on the smooth surface. And another one is the sex pili. Now, sex pili is complete. Is a little bit different for their structure of the other ones. Okay, so the sex pili, okay, so this is sex pili, is responsible for the exchange of the genetic materials. Okay, it's said to be a conjugations. Oh, sorry, the conjugation. Okay, so I described the conjugation mechanism in lecture 3. Okay, so they often found in the negative gram state. So this is a picture showing you the conjugations. So the plasmid is transferred from one to the another through the sex pili. So the sex pili is longer, it's much longer, okay, than the common pili. So this is easily to be distinguished for the sex pili, okay. So the fourth one is a characteristic of the bacterial cell wall, okay, it's a gram walls, okay, it's a gram staining, okay. So because the peptide gigands only present, okay, on the surface of the bacteria, okay, as a major component, but not all, okay, the bacteria can be stated. For example, the mycoplasma. So mycoplasma, why? Because they have a wax. There's a wax on the surface. So we need an acid to dissolve it, okay, and then we stain it. So they have a different color. So it's a fast staining procedure instead of the gram staining, okay? So there are two groups, okay, as I described it earlier. The first one is gram-positive. They're blue-purple because they are very large, they are very thick of the peptide ligand. So they resist, okay, to the enzyme. So they resist to enzymes. So the color, okay, the color is the purple. It's blue-purple. But gram-positive because the peptide ligand is in between the outer membrane and the plasma membrane, so it's easily the enzyme, okay, enzymes, okay, can be colored in the red color, okay, so it's a blue, red, or purple or red color, so it's easy to be distinguished, okay. So to remind you again, okay, the gram-positive, gram-purple, in which the stain color is blue or purple, and gram-negative is red or pink. So this is the mechanisms, okay, of the methods, okay. So the bacteria is stained with the first step. So I will show you the video, okay, after the description of the brief of this slide, okay. So firstly, the bacteria they cultured, and then they form a smear on the surface of the slide, and then we stain it with the crystal violet. It's a purple color, okay, because, okay, the thick of the wall of the gram-positive, okay, so they will stain it all in the purple color, and then we fixed it with the formations, okay, and they stained it with color, with water, and they didn't come out, so they're gram-positive, and stain come out in gram-negative form. So there's two color, so these are colorations, okay, done in the gram-negative and the purple color in the gram-positive, okay. So this is very easy. It's a straightforward of the staining. It's not that long time. So I'll show you the video, okay, on about the gram staining procedure. So let's take a look for the gram staining procedure now.
The gram stain is the most widely employed staining method in microbiology. It is a differential stain because it divides bacteria into two classes, gram-positive and gram-negative. In the first step of the gram stain procedure, cells from a fresh culture are transferred to a clean slide and allowed to dry. If the cells are on an agar plate, they should first be transferred to a liquid medium for dilution. The cells should form a thin, barely visible film. This can be achieved by smearing cells obtained from the surface of an agar medium or from a liquid culture. Fresh cultures must be used because as cells age, they lose their ability to retain the stain. The cells are then fixed to the slide by passing slightly above the flame of a Bunsen burner. After passing above the flame, the slide should feel warm when touched to the back of the hand, but should not be too hot. The fixed cells are then stained with the basic dye, crystal violet, for 30 to 40 seconds. The slide is then rinsed with water to remove excess stain. At this point, all cells appear purple under the microscope. Next, a solution of grams iodine is added and retained on the slide for about 1 minute. The iodine combines with the crystal violet to form a di-iodine complex, thereby decreasing its solubility within the cell. At this point, the cells still appear purple. The cells are then decolorized by washing with ethanol or acetone. This is the differential step. Gram-positive bacteria retain the crystal violet, whereas gram-negative bacteria do not. The ethanol or acetone should be added drop-wise, with the slide tilted at an angle until the drop coming off the edge of the slide just starts to become colorless. Even gram-positive cells can lose the crystal violet iodine complex during prolonged excessive decoloration. Excess ethanol is then washed off with water. When viewed under the microscope, gram-positive cells appear purple and gram-negative cells are colorless. Finally, the rinsed cells are covered with the counterstain safranin for 20 to 30 seconds. This stains the gram-negative bacteria pink. After rinsing with water, the slide is dried with filter paper. When viewed microscopically, the gram-positive bacteria are purple and the gram-negative bacteria are pink. Generally, the gram stain correlates with the cell wall structure among the bacteria. The ethanol is thought to shrink the thick peptidoglycan in gram-positive cells, thus retaining the dye. The thick, dehydrated peptidoglycan layer of gram-positive bacteria appears to be a permeability barrier, preventing the loss of the crystal violet iodine complex. In contrast, the peptidoglycan in gram-negative bacteria is very thin and has large pores. Ethanol may extract lipids and increase the porosity, thus removing the crystal violet iodine complex.
Okay, so let's take a look for the lecture here. Okay, so this is about the grain staining methods. Okay. Right. So now there are two categories. Okay, of the groups of the bacteria. So there's a gram-positive coli. So this is how they. So these are the medically important groups of the bacteria. Okay. So the gram-positive and gram-negative. So these are the examples of the gram-positive coli bacteria, and these are the gram-positive rod. Okay, the rod-shaped bacteria, and the right-hand side is gram-negative coli and gram-negative rods. So for the next lectures, I will tell you some of the profiling of the bacteria. For example, the Staphylococcus coli, okay, E. coli, and etc. So, and as I said earlier, some of the bacteria, they do not have cell wall. For example, the microbacteria, okay, tuberculosis. So they can't be stained. They can't be stained by the gram staining. So the only way is they use the acid fast stain. Okay, so the color look like the red color. So these are the slide of the bacteria. Right. Now let's take a look for the respiration. Okay, so the bacteria, because as I said, the bacteria, they have no choice. Okay, they can't walk from one side to another. So they can't move. So they need to adapt of the different environment. So the bacteria, they localize in one certain area. But of course, in the modern world, because of the transportations, so the humans or the species, they bring the bacteria to move from one place to another. Okay? But mostly the bacteria only localize in a certain area. And that area has a different range of the oxygen tension. For example, some bacteria found in the Himalayas mountain or Everest mountain, the oxygen level is very low. Or some bacteria is found in the underneath of ocean, the sea, the watery, so the lack of oxygen. So some bacteria, they used it to be adapt in the very low oxygen in the Himalayas mountains or under the sea or in the air here. So they're classified in 5 groups, depending on their oxygen requirement. Okay, the first one is obligate. They require oxygens. Atmospheric means, what is atmospheric? About the air. Okay, so in the air, okay, about 71% of nitrogen and 20% of the oxygen. So they need an air oxygens. So they found in the air. So the microbacteria is a good example because they usually found in the lungs. Okay, the lung is about the air. The atmospheric air pressure is similar to humans to the outside of the environment. Okay, so the second one is the microaerophilic. So philic means they love it. They love it. Okay, they only require small amounts of the oxygens. Okay, so they can found in the anywhere. For example, in a room, there's a corner of the room, there's some dead air, or some very low oxygen in the corner of the air. So they need oxygens. I need oxygen, but they can need very small oxygens. Okay, so they can live happily in 21% of oxygens, but they also they can be exit is a very low oxygen level. So the low oxygen level, they can make them happy. So this is meaning of the micro. Micro mean little. Okay, they love a little air. Okay, so the third one is the facultative. The facility. The facultative means they are capable to live with a very low fluctuations of oxygens from 0 to 21 oxygens. They are usually found inside the human body in various parts because in various side, intestine or the blood, okay, or some tissue, they have a different range of oxygens. So they are flexible. They can survive. They're capable to survive in the different range from 0 to the air, to the atmospheric air or to the oxygen. So they can live. They can survive. So this is very common bacteria. So when you look at here on the right-hand example, the enteric bacteria. Enteral means the bacteria found in the intestine, okay, in the gastrointestinal tract, in the digestive system, the most commonly found because the different range of the oxygens, okay, the most commonly found. Or aerotorrents. So aerotorrents means they live in the lack of oxygens, but okay, so they do not require oxygen. Okay, I don't need money. Okay, but and they grow better, but they can live, but not happy. So they don't like money. Okay, they don't like cash. Okay, I like live without any cash, but if you give me any cash, okay, I can accept that. So this is about the aeroturbulence, okay? So they don't need oxygen, but if they have, okay, I get it, okay? And final is the obligate. Obligate, they only live in the absence of the oxygen. They live without any oxygen. If they give oxygen, they die. So they are the extreme of the bacteria. So they're extreme group of the bacteria. So they're not happy with they're not happy with the oxygens. Okay, if I give you oxygen, they die. Okay, so these are the examples. Okay, so let me summarize this group of the requirement of the respirations. The first group is they need money. Like me, I need a lot of money. Okay, so the first one is the obligate. Absolutely, oxygen is needed. Like me, I need money. If I have no money, I can't survive. So yeah, we are obligate of the bacteria, not obligate aerobes. Okay, aerobes mean they extremely need oxygen. And second one, micro aerobic philic bacteria, they leave a little bit money. So I can have different, I can survive with different amounts. Okay, salary, for example, $10 or $100. Okay, I can live with it. The fluctuative is a range. They can survive 0 to 21%. They're still happy. Okay, I have no money, I'm happy. I have full money, I'm happy. Okay, so the fluctuative aerobics. And aerotolerance means I don't need money. Don't give me money. I don't live. Okay, I live without any money. I'm okay. I live in jungle. Okay, I'm one man alone in planet. I don't need money. If you give me money, okay, I get it. All right, okay. And the final one is the obligate anaerobics. So the obligate anaerobes means don't give me money. No, I don't like money. Don't give me money, right? I don't need oxygen. If you give me oxygen, I'll die. Right, so there are 5 groups of the bacteria. So that's it for the 5th point of the respirations requirements. So the bacteria have different requirements of the air and the modes of the nutrition. Okay, so they exhibit different modes of nutrition levels. The first one is the autotropic. Auto means automatically. Me is like me, auto. Okay, I can find the food. I can hunt it. Okay, so this group of bacteria, they can make their own food. Okay, like a plant, they can synthesize. Okay, so synthesize the food, so they can convert the inorganic materials into organic materials. For example, carbons, hydrogen, oxygen, so they can convert the carbon, hydrogens, and oxygens and convert into the CHO, the carbohydrate, the food, the sugar. Okay, so for example, phototropic. So some of the bacteria, they can use sunlight like a plant. They use the photo. Photo means the sunlight to make the food, or synthetic bacteria. So chemo means the use of the chemicals to make the food. Okay, the autotrophic. But mostly are heterotropic. Hetero means they need to obtain the food from others, like humans. We can't make the food. So for the human, we need to hunt it, the food, or we give some money to buy the food to get it. Same as bacteria. They need to hunt. They need to get the food. But they are unable. So this group of the bacteria, they are unable, okay, to make the food. Okay, they need to obtain the organic food. Okay, so and good example. So most of the bacteria, almost all bacteria, they are saprotropic. Sapro means they rely on the decompositions of the food. So what is decompositions? Okay, like a human diet, they die. The human body, the enzyme in the human body will degrade, will dissolve the tissues. So we decompose it. So the organic substances is the enzymes catalyzed. It's catalyzed by enzymes. So the tissues lost their integrity. So the organic is back to the inorganic. So carbon, hydrogen is okay. So the organic, for example, protein is degraded in peptides. Okay. So the bacteria, they don't have mouth. So they will release the enzymes, okay, to decompose it, the organic substance, and then absorb it, okay. So that means the saprotropic, okay. is rely on the decomposite of the food, decaying of the substances. The third one is about the last two, rows of bacteria. The first one is symbiotic. Symbiotic means they have a mutual win-win. Now, on the human surface, in my lecture notes, in the lecture 4, in coming AMP2 lecture 4, when we talk about the defense system, now the skin, on the surface of the skin, there's about 500 or more of the symbiotic bacteria. They don't harm us if we are healthy, so they will get the food. They will fight with the other colonies, so they are healthy. That benefits to other species. For example, the nitrogen-fixing bacteria. So what is this? Nitrogen. So as you remember in your secondary education in biology, the plant, they need 3 elements: the potassium, nitrogen, and the phosphorus. The plant, they need amino acids. So what is the major component amino acid? Nitrogen. However, nitrogen is mainly in the gas form in the air. So how to get the nitrogens into the solid form then? By the air, from the storm, of course. Can you have so how frequently and how often of the thunderstorm happen? Very seldom, right? Rare, So they need bacteria. So the roots, the roots of some of the plant, the roots, they will contain some bacteria, so they can fix the nitrogens. They can fix the air nitrogen into carbon form. So the nitrogen can be combined with the hydrogens to form ammonia. Remember the ammonia, the nitrate. So these become the nutrients for the plants. So the nitrogen is fixed, from the air into the nitrate forms and ammonium form degraded. So this is the help of the bacteria. So the nitrogen fixation bacteria, or the ammonia, is removed. For example, for the water treatments, the sewage is smelly because of ammonia. So the nitrogen is removed, from the solid form, the ammonia, into air. So they need a denitrifying bacteria. So there are 2 groups of bacteria, to responsible for the nitrogen cycling of nitrogen elements, from the inorganic to organic form and organic back to inorganic form by 2 sets of bacteria. So they're beneficial. They're beneficial for them, and benefits to the environment or beneficial to the host. So these are the symbiotic bacteria. However, some of bacteria, they're harmless, they're pathogenic. For example, E. coli, as I said, in the cholera, the vibrio cholera, make us a serious diarrhea or make us the inflammation of brain. So they're parasitic. So what's the meaning of parasitic? It means they will harm us. They rely on hosts. The bacteria rely on hosts, or plants, animals, and humans, but they will harm the humans because they really need us. They feed on us, but they feed on hosts and will harm us and make us sick and even die. So, and all the pathogenic bacteria are parasitic bacteria. So that's easy. So there are 4 nutritional modes of bacteria. Now about the reproductions, so all the bacteria, they perform into the binary division. So what is a binary? It's 1 to 2. It's a 2 to power n. It's a binary. Binary means 2. 0, 1 is a computer language. So 0, 1, 1, 0, and then etc. So it's a 1 become 2, 2 to 4. So it's follow the 2 to n power. So it looks like the human cell division. It's like the human division, but the time is much shorter, about 9 minutes per cycle. So 1 cell into 2 identical offsprings. So this is the way how they split it and how the continuous of their species. Right. So let's take a look so these are the trees, of the gram-positive bacteria. And to summarize the table about the classifications, as I said, the first classification is the profiling. So this is look at profiling of bacteria. For example, the shape, the rod shape, the spherical shape, or the filament shape, or the spherical shape. And then they further divided into aerobic or anaerobic, or into 5 subgroups of the, for example, the obligate aerobics, obligate aerobes, okay, the aerophilic, okay, microaerophilics, okay, bacteria, facultative aerobes, the aerotolerance anaerobics, and the obligate anaerobics. So the 5 subgroups of the requirement of the respiration, and further, or some of the classification is to classify amount of their modes of nutrition. Okay, so that's it for about the classification system. Now remember, in our first lectures on the classification, the taxonomy of the kingdoms. So there are 3 types of the classifications. Okay, for example, the 2 domain, 5 kingdoms. Okay, and then 3 domain, 6 kingdoms. Okay, in the latest one, there are 3 domain and 6 kingdom. Okay, the Monria and Acharia. So Acharia means ancient. It means they were, because ancient, they are buried. For example, they're found in, usually these kinds of the bacteria is found in some extreme, okay, or underneath of the soils. Okay, they are not found. Okay, so they are and then they discovered by recently, so they name it, and they have and they are, so they, and especially they are not really looks like bacteria. Okay, so Acharia is archaeology, is a term, is a suffix, means they are ancient, okay, species of bacteria, and they are originally found ages ago. Okay, so these are evolved, they exist more earlier than bacteria. So why the name is Acharia, is a bacteria, or is lived, okay, earlier than bacteria. So they combine these 2 words together into Acharia. And they have the 60S. Remember, 60S RNAs is found in bacteria, not in humans. The 60S bacteria only found in the Acharia. But more interestingly, they're more closely related to the eukaryotics. Okay, they have lack of the true nucleus, but the RNA shown that they're more related to eukaryotics. It's quite confusion. So they separate them from the bacteria into one single kingdom and one single domain. Okay, is about the Acharia domain. Okay, and Acharia is an ancient bacteria. So it's there's a lot of reports founded, okay, in the Yellowstone, or in the volcanoes, or in the other areas. Okay, and then they collect. It's a rising numbers of the bacteria. Okay, of the Acharia. Okay, and so far they're not associated with the disease of human and animal reports. Okay, so this is one. This is distinguished from the bacteria. Okay. Now so as I said, so these bacteria, they're found in extreme place. Okay, volcanoes. No one will live in a volcano, so they're not really associated with humans logically. Okay, so there are 3 kinds of the Acharia. The first is the thermophils. They love, phil means they love the thermal. Thermal means they need to live in extreme temperature. Okay, the temperature more than 45 degrees Celsius, because on average no human can live very happy more than 45 degrees Celsius. No, you can't survive it. So mostly in volcanoes. Or halophils. Halophils means they live in extreme salty conditions. Okay, or mephogens. Okay, these are large groups. Okay, so because they're commonly found in the wetland, because the marsh gas. Okay, when you get hiking, you experience a gas, unpleasant smell, so marsh gas. Okay, because this group of Acharia can produce the metabolics, mainly methane. So methane is one of the metabolites. Okay, byproducts from the methanogen. So some people experience some of the unpleasant gas. Okay, and these are toxic gas. Okay, so these are 3 conditions. Okay, the condition of the production of methane, or in a very high salty condition, or extreme temperature conditions. So this is methanogens. Okay, so this of the bacteria, they can produce the methane. Okay, that's why the methane produced in the cow or the cattle. Okay, and that's one of the factors, okay, contribute to the and on the global warming. Okay, so that's it for today's lecture. So hopefully, so these lectures, this stream live lecture will be saved, okay, and will be kept on the public until Friday. And then I will serve the links, okay, and upload and then attach the links to this show, okay, so you can watch the replay of these lectures, okay, via the show link that I provide you. Okay, thank you for watching and thank you for attending the lecture today. Thank you.